work at the BHSAI, which is a computational biology research institute funded by the government. We're located in Frederick, Maryland. And today I will be talking about how to control inflammation. Of course, we all know about inflammation from our everyday lives. It's the redness and the swelling and the pain. But what I want to emphasize right off the bat here is that inflammation is controlled by a very complex biological system. Now, this cartoon in the slide is a gross oversimplification, but it shows that in response to injury, the body mobilizes an array of inflammatory cells, such as neutrophils and macrophages, that can exist in a variety of phenotypes and that perform a number of functions at the injury site. For example, they ingest cellular debris, which resulted from the injury, and they release a large number of molecular mediators, many of which are proteins, such as, for example, TNF-alpha. And the role of these mediators is to coordinate the inflammatory response. So together, the inflammatory cells and the mediators participate in a large number of processes, such as molecular mediator production and release, cell phenotype conversion, and cell migration. The physiological role of inflammation is to facilitate wound healing and promote tissue regeneration after injury. However, in certain cases, inflammation can become pathological, meaning that something is malfunctioning in that complex system that we're talking about. And when inflammation is pathological, that actually can lead to impaired wound healing. Moreover, pathological inflammation can contribute to such serious diseases such as cardiovascular diseases and cancer. So how can we distinguish between physiological or normal inflammation and abnormal or pathological inflammation? We can do that by looking at the inflammation time course. If we plot the concentration of inflammatory cell or a mediator over time, then in the case of normal inflammation, we will obtain this characteristic one-peak shape, which shows that inflammation intensity increases, reaches some maximal value, and then it decreases or resolves, going to some baseline. However, in the case of pathological inflammation, this time course is distorted. For example, all the levels of inflammatory regulators can be strongly elevated, which is known as the cytokine storm, which in fact can lead to systemic inflammation and sepsis. Another example, chronic inflammation. In this case, the peak of the inflammation time course looks fairly normal. However, the tail of the time course is elevated. And in this case, normal inflammation resolution does not occur. So from these examples, you can see that it's really all about the shape of the inflammation time course. And to analyze such shapes in a systematic fashion, researchers came up with 4 quantitative parameters called inflammation indices. 2 of these indices characterize the timing of the inflammatory response. For example, resolution interval tells you how quickly inflammation will resolve. And the remaining 2 indices characterize the intensity of the response. For example, the peak height shows you the maximal level of the inflammation time course. And this brief discussion about time courses brings us to the main question of my presentation. How can we control the inflammation time course? How can we modulate these inflammation indices in a desirable way, and can we do it in a selective way so that we control some of them but not others? And most importantly, how can we shift the time course for pathological inflammation toward the time course for normal inflammation? So once again, just to emphasize, these white arrows in the slide, this is what we want, this. So how do we achieve it? Well, our approach was based on a very simple idea, and that idea is that we could perhaps control inflammation time courses if we identify the processes that strongly influence inflammation time courses, and then we can modulate those processes. In fact, I already mentioned some of those processes earlier in my talk, but in reality there are dozens of them, if not hundreds, and they interact in very intricate ways. Moreover, the inflammation time courses can be very variable. They can be diverse based on different factors such as intra-subject variability and perhaps the wound location and severity. So we really need to have a very efficient way to screen potentially thousands of inflammatory scenarios if we want to analyze the impact of different processes. And this motivated us to use an approach based on computational modeling. We developed a computational model of inflammation, which is essentially a system of differential equations. And the model has 18 variables. These variables represent essential inflammatory cells and molecular mediators. The model has 69 parameters. These parameters represent the rates of 57 molecular and cellular processes, which means that we can assess the contributions of different processes to the inflammation time course by determining the influence of different model parameters on different model variables. So here's a brief outline of our computational strategy. We developed the model, we calibrated and validated it using published data, and then we used our model to perform 10,000 simulations with randomized parameter sets. Every simulation represented 20 days of inflammation after injury, and for every simulation we calculated the time courses for all the model variables. And then we use this data to calculate the 4 inflammation indices for each time course. So we have the inflammation 4 indices for each model variable for each of the 10,000 simulations. And finally, we use this information to perform sensitivity analysis and correlation analysis. In the sensitivity analysis, we perturbed the model parameters one at a time. We calculated the resulting change in the inflammation indices for all the model variables. And then we divide the perturbation in the index by the perturbation in the parameter, and that's our sensitivity. In the correlation analysis, we calculated correlation coefficients between each model parameter and each inflammation index for each model variable. Together, these two methods gave us the information that we used to select the most influential model parameter for which we could use for further analyses. So just to give you a flavor of what the results look like, here I'm showing you our results for the macrophage peak height. The pie chart in the slide represents the 10,000 simulations. And what this tells us is that in 89% of the 10,000 simulations, the single most influential parameter affecting the macrophage peak height was the rate of the macrophage influx into the wound. And in the other 11% of the simulations, this most influential parameter was some other parameter in the model, depending on the simulation. As another illustration here, I'm giving you the results for the resolution interval for macrophages. And we can see that in 35% of the 10,000 simulations, the single most influential parameter affecting macrophage resolution interval was the rate of macrophage efflux out of the wound. In another 10% of the simulations, this most influential parameter was the platelet degradation rate. And in the remaining 55% of the simulations, it were different parameters in the model depending on the simulation, but none of them were as prevalent as the first two. So these results clearly show that according to our computational strategy, macrophage efflux and macrophage influx can very strongly influence the inflammation time course. Correlation analysis gave us some additional insights into inflammation regulation. And here I'm showing you the results for the peak height. On the x-axis, we have the model parameters. On the y-axis, we have the model variables for which we calculated the peak height. And the black rectangles in the figure show you the situation where the correlation coefficient between the model parameter and the model variable exceeded or was at least 50%. So here we can see that the macrophage influx rate was strongly correlated, in fact, with several model variables for which we calculated the peak height, not just with the macrophage abundance. this actually extends the results of our sensitivity analysis. And another interesting finding was that the TNF-alpha degradation rate was strongly correlated with the height of the TNF-alpha peak. And because TNF-alpha is such a prominent inflammatory regulator, which participates in many different processes, this result tells us that the TNF-alpha degradation rate is quite likely to affect the inflammation time course very strongly. Here's another example. I'm showing you the data for the resolution interval, and here what I want to point out is that the CXCL8 degradation rate was strongly correlated with the resolution interval for active neutrophils in the wound. CXCL8 is in fact a chemoattractant for neutrophils. And we know that the abundance of active neutrophils in the wound is one of the strongest initial drivers for inflammation. This tells us that the CXCL8 degradation rate is likely to be a strong modulator of the inflammation time course. After we obtained these results for sensitivity analysis and correlation analysis, we performed some additional filtering. And as a result, we put together a list of 5 processes: macrophage influx, macrophage efflux, TNF-alpha degradation, CXCL8 degradation, and TGF-beta degradation. Our computational strategy predicted that if we modulate these processes, then the inflammation time course will change. But can we shift the time course for pathological inflammation toward the time course for normal inflammation if we modulate these 5 processes? We chose to address this question for 3 out of the 5 processes, specifically the ones here that involve TNF-alpha, CXCL8, and TGF-beta. All of them are protein mediators of inflammation. And we made this choice because we knew a practical way how to modulate these processes. We can do that by using protein inhibitors. Actually, we can reflect that inhibition in our computational model if we incorporate the relevant biochemical reaction into the model. That's exactly what we did. And here I'm showing you the results for neutrophils. So the green curve in the slide is the time course for normal inflammation. The magenta curve is the curve for chronic inflammation. And you can see that in the case of chronic inflammation, the peak is elevated, so we want to push it down. And the tail of the temporal trajectory is shifted to the right, so resolution is delayed. But then potentially, we want to shift the tail to the left. So the blue curve corresponds to chronic inflammation with an added TNF-alpha inhibitor. And you can see that this inhibition pushes the peak height down. However, it does not affect the tail of the inflammation time course. And this tells us that TNF-alpha inhibition potentially can be a strategy to selectively modulate one inflammation index, the height of the peak. In contrast, if instead of inhibiting TNF-alpha, we inhibit CXCL8, we see that the peak of the inflammation time course does not change. However, the tail of the inflammation time course is shifted to the left, which means that the resolution time for inflammation is reduced. And in fact, in this case, it is quite close to the resolution time for normal inflammation. So this is a strategy to selectively modulate the time of inflammation resolution. And looking at these results, one natural question that comes to mind is, can we modulate both the peak height and the resolution time at the same time? Well, these results suggest that perhaps we should inhibit both TNF-alpha and CXCL8 simultaneously. This is exactly what we tried in our model. The result is in the slide, and we can see that indeed the peak height goes down and the tail of the inflammation time course is shifted to the left. So if we put together everything that I told you today, we can come to 3 main conclusions. The first conclusion is that we identified 5 processes that significantly impact the inflammation time course. The second conclusion is that inflammation can be strongly modulated by inhibiting TNF-alpha, CXCL8, and TGF-beta. And the third conclusion is that simultaneous inhibition of TNF-alpha and CXCL8 can reduce both the inflammation peak and time to resolution. And I would like to add that we are currently collaborating with an experimental group to test these computational predictions in a mouse model of chronic inflammation. Now I would like to thank the people who contributed to this project. Sridevi Nagaraja is the BHSAI scientist who actually did this computational work. Jacques Reifman is the BHSAI director who made critical contributions to the study design and data analysis. And I would like to thank our funding agency, which is the Clinical and Rehabilitative Medicine Research Program of the U.S. Army Medical Research and Material Command. Thank you.